Okay. So, hello and welcome everyone to the week 6 live session course on experimental biochemistry. Today we would be discussing about in brief, the topics that were told to you in week 6, and then we would be talking about the assignments and a few question and answers. Okay, so let us start with the lecture. I'm sharing my slides with you all. So I hope my screen is visible to you all, and so I hope my screen is visible to you all, and I will be sharing my slides. So in today's class, we would be mostly discussing about a few very interesting, or rather more of lab things, right? So you basically have heard of electrophoresis, yeah, the SDS page electrophoresis, agarose gel electrophoresis. So today's live session will mostly cover these areas. So let us first introduce to you what gel electrophoresis is. So basically, gel electrophoresis is a technique used for the separation of DNA, RNA, protein molecules using electric current applied across a gel matrix. So from the term electrophoresis, you can already understand that it refers to the electromotive force, or the EMF, for the movement of the molecules across the gel matrix. Now, upon placing these molecules in wells, be it your DNA molecule or an RNA or even a protein, so for that you have different types of gel electrophoresis which you can use. But at the end of the day, what you need is you need to apply current. You need to load the samples in a gel, apply current, and the molecules will move at different rates. You watch it. Now this the separation of these molecules, or the movement of these molecules, is based upon charge to mass ratio towards its movement towards a negatively charged, if it's negatively charged, it moves towards the anode. If it's positively charged, it moves towards the cathode. So the mobility is dependent on various factors. Sometimes it can be according to charge by mass ratio, sometimes only mass, sometimes the complexity of the structure of the molecules that we want to check the electrophoresis using. Okay. So the principle behind electrophoresis is that we need to understand how the gel apparatus is connected to the power supply based on Ohm's law. So you have the voltage, which is equal to V equals to IR. So here you have to consider the three factors. One is voltage, current, and the resistance. So the gel present over here can be viewed as the resistor, and the power supply as the voltage and the current source. So most power supplies deliver constant current or a constant voltage, and also delivers a constant power, because power is equal to voltage into current. So a constant power is very important for the movement of the molecules upon loading it on a gel matrix. Now, if you look into a commercially available equipment of or a gel apparatus, what you would be seeing is that the positive terminal is colored red and the negative terminal is colored black. Now, this configuration is designed to work with a vertical slab gel electrophoresis, or even a horizontal gel electrophoresis for a protein sample, and the negatively charged proteins or the nucleic acids moves towards the positive electrode in the low buffer chamber or an anionic system. Okay. Now, when a single gel is attached to a power supply in an anionic system, the negative charges flow from the negative cathode, that is the black terminal, into the upper buffer chamber through the gel and the lower buffer chamber, which is connected to the positive anode. The red terminal that completes the circuit and that allows the constant voltage followed by a constant current to be flowed across the gel matrix, that allows the complete circuit to lead to the mobility of the molecules according to the factors that are involved over here. Now, a very interesting system of this gel electrophoresis is the page. So most of you have heard about page. So what is page? The full form is um, po- polyacrylamide gel electrophoresis. That's an anionic system because of the negatively charged SDS. We call it the SDS, page sodium dodecyl sulfate polyacrylamide gel electrophoresis. So it is because of the negatively charged SDS molecules present over there, and these proteins are separated in the cationic systems, and that also very occasionally, if you are using some kinds of detergents or reagents that can positively charge the proteins over there, you can use it. Now in these gels the proteins are positively charged because of, now what happens in the cationic system is that the proteins are positively charged because of the pH of the gel buffers or due to the presence of cationic detergents, as I was saying, the CTAB or the cetyl trimethyl ammonium bromide. Now, proteins move towards the negative electrode, that is the cathode, and the polarity is reversed paired to the SDS page. So, in normal SDS page, the molecules or the proteins move towards the anode because they are negatively charged, but in a cationic system, it moves towards the cathode because the protein is positively charged. Okay, so but now when we know about SDS page, we also need to know about an agarose gel electrophoresis, or AGE. That's the age. So what is this agarose? So it's an extracted from the seaweed and is a linear polymer of sugar molecules, you can see over here. Now when heated in water or any kinds of buffer, the agarose polymers are flexible and the mixture of agarose and water liquid. Now when we cool it, again these linkages start forming, the polymers start forming a matrix, the mixture becomes more solid and gel like. Now the concentration of agarose determines how dense the matrix will be. Now if you are using more amounts of agarose, uh, the holes or the blank spaces, the complexity of the matrix will be low for a lower concentration of agarose. If you're using higher concentration of agarose, there will be less places, the gel will be more dense, thick, and that allows for the separation of small molecules. Like for example, the higher the concentration of the agarose that you're using, it will be for separating small molecules. Same, the concept is applicable for an SDS page, an SDS polyacrylamide gel electrophoresis, and a similar concept is there for agarose. So for resolving higher molecular weight or higher base pair DNA or RNA, you need to run lower concentration of agarose gel, and for resolving a lower molecular weight or a lower base pair wala DNA and RNA, you need to have a higher concentration of agarose for the better resolving. Now this is it is the agarose gel electrophoresis. So you see the red is connected to the positive terminal and the black is to the negative terminal. So your negatively charged DNA molecules will move towards the positively charged or the anode. This is the basic power supply, and this is the apparatus in which you put the gel. You load the samples in these blue wells, and you run the gel constant power at a constant power supply. Now we need to compare these gels. So how do we compare the DNA fragments? So these fragments separate by size. So as I was telling, if you are loading a DNA or a RNA, for example, for an agarose gel electrophoresis, we usually do that. So the molecules or the nucleic acids will separate according to the number of base pairs. Okay, so higher the base pair, it will be running at a higher are very close to the wells, and the mobility is faster for the lower sized or the lower base pair wala nucleic acids. So how do we separate the DNA fragments? We run it through the agarose gel and we carry out gel electrophoresis, and then we check it. Now, gel electrophoresis is nothing but a method of separating DNA in a gelatin-like material or an agarose gel, you can tell, using an electric field. So, DNA is negatively charged. When an electrical field is applied, or when you apply a constant power supply, it moves towards the positive side, depending on the you say it's the number of base pairs that it contains. So you load your sample over here. This is the negative terminal and this is the positive terminal. So the complexity of the DNA molecules determine whether its mobility will be fast or slow. So you can tell how complex your DNA molecule is, as well as what is the length of the nucleic acid. Now DNA moves in an electric field. So how does that help you to compare the DNA fragments? Now the size of the DNA fragments affects how far it travels. Smaller pieces travels faster, larger pieces travel slower, and it lags behind. So that's simple. That's simply as swimming through the jelly-like material or the jello, where the jelly-like material is your agarose matrix, or even a gelatin-like material. Okay, so what happens over here, you have a DNA and you do some kinds of restriction digestion. So what do, you digest your DNA using some kind of enzymes, endonuclease or exonuclease enzymes, and then you want to check it in a gel. You run the gel, it runs at the different positions, and depending on that, the longer fragments lags behind and the shorter fragments runs faster. And then when you look at it under UV transilluminator, you can see whether you can see these glowing bands under the transilluminator, and from there you can tell whether the restriction digestion process has successfully occurred or not. So that's mostly about agarose gel electrophoresis. Okay, so you have a mixture of DNA molecules of different sizes in this particular sample. You load them onto the gel, and you allow the power supply to take over it. So you cut and then you cut the DNA into different with different restriction enzymes, and then you check it. You will be observing different bands over here, and you can compare using the sizes after the restriction digestion has occurred. So you can see a lot of things over here in a gel when you are doing restriction digestion, or even when you want to run the intact plasmid. So usually in an intact plasmid you observe 2 to 3 bands because of the circular because of the CCC circular supercoiled structure. You have the even the linear form and more of the circular form. So there are 3 forms that where your plasmid does exist. So in a normal plasmid you observe 3 bands, but when you're doing restriction digestion you see more than 3 bands or multiple bands, depending on how many restriction enzymes you have used, or you can say the number of types of cuts you have introduced inside your plasmid. So there are 2 ways of how you can stain the DNA. The easier way is to just you put it under a UV transilluminator and you click a picture of it. You are very well to see, because when you are running the gel or preparing this agarose gel, you use some kind of ethidium bromide. So you use a less amount of ethidium bromide. So ethidium bromide intercalates among the major group of the DNA, and it allows the DNA to glow up under the UV transilluminator. So it's like a fluorescence. Okay. So you can see that when okay, I will do. Yeah. So that happens over there. Okay. So when you can do that using ETBR, or the other way around is Carolina blue stains of DNA. That also you can use for staining the DNA for long-term purpose, like we do for SDS page. We have Comasi blue. We do it for DNA. We use the Carolina blue stains. Okay, so far well and good. Now, the question that you were asking is, what is a marker DNA? Now, you know that your plasmid is probably of a size of 6,000 base pair. Okay, you know that. But after restriction digestion, probably there are several cuts that have been introduced inside your plasmid. You need to compare them. How do you compare them? With whom do you compare them? That is the place where your marker DNA comes into play. Why is it so? What is this marker DNA made up of? This marker DNA is made up of several base pair sizes of our DNA material. What does that mean? So the marker DNA is nothing but a molecular weight indicator. Okay, what does it indicate? So this comprises of several sizes of DNA put in a particular sample. So for example, you have 100 base pair, 200 base pair, 500 base pair, 1000 base pair, and so you prepare DNA of that particular length, because you are synthesizing a DNA. So you can make it up to 100, 200, 500, you know it. So you make this DNA, put it in a particular vial, give it a color, and you run it. What does that do? So when, if your DNA of your particular desired DNA is of size, say 150 base pair or say 1500 base pair, you can compare the size by looking at the molecular DNA. So maybe this is 100, this is your 200. So if your DNA is 150, it should come in between this. You can tell that yes, so my cut has occurred at the right place, and if it's cutting at 1500, you're supposed to get it somewhere like this. You can tell that yes, this is the also the desired region, because this marker DNA is probably of the size of 2000 base pair, right? So that is the molecular weight or a base pair indicator that you always run whenever you're running an agarose page or an SDS page. That gives you an indication of the correctness of your cut size or the size of the DNA that you want to achieve. So it gives you, and so it's a reference as such. Okay, now yeah, both are the same. So it's a ladder, or the marker is both the same. You give it different names, like I was telling, molecular weight indicator, the reference indicator, you have a ladder or something like that. So it's same, it is just for used as a reference. Okay, great. Now, so that is all about your agarose gel electrophoresis. The other type of electrophoresis, probably you people have done it, so it's the polyacrylamide gel electrophoresis. It's an SDS page apparatus where this is the power supply, you connect it to the two ends over here, and you run a gel. And this SDS page apparatus comprises of several components which we use. So let us start from the first one. So this is the cassette, or the apparatus in which you pour the buffer, you have the gels loaded onto this, and then you run it. But the way we should start it is basically from number 7. Why is it so? So we need 7, 5, so from 2 to 7, all of these is used in the preparation of an SDS page. Why is it so? So this is the cassette where we prepare the gel for the gel preparation, and this is the holder of the gel plates. So this is a short plate, this is the wide plates, or the thick plates. On we clamp on both these two, and we tighten it using these clamps, and we load it onto it for the preparation of the gel. And there are several kinds of wells present over here, the 10 well gels, 15 wells, 5 wells, and there can be multiple ways of depending on how many samples we want to run as such. Okay, so depending on that, we prepare the gels over here, we load onto it, and then we run the gel. So after running the gel, we stain it using, there are multiple ways of how we can stain it. We have the Kumasi blue stain, we have the silver staining, sometimes fluorescent staining and all. So we can use all of these depending. And so number 6 and number 1 is basically your marker or the ladder. So reference protein ladders we have, which we load for checking of the molecular weights of the protein. So probably we can tell it is of a size around 95 kilodaltons or such, because the lower band is showing up like that. So maybe 100, so it can vary between 100 to 95 kilodaltons. So you see this after you stain the SDS page. Okay, so now we need to know something about the separation so of an SDS page, so or even an agarose gel electrophoresis. So from the term gel, we can tell um, And the term gel in this instant refers to the matrix used to contain and separate the target molecules. In most of these cases, the gel is a cross-linked polymer whose composition and porosity is chosen depending on the specific weight and the composition of the target that we want to analyze. For example, if we want to, like I was telling, if we want to resolve a smaller molecular weight protein, we can use a higher composition of polyacrylamide. If we want to use a lower molecular weight, for resolving a higher molecular weight protein, we use a lower amount of polyacrylamide. For resolving a lower molecular weight protein or a sample, we use a higher concentration of polyacrylamide or an agarose. Okay. So now when we're separating proteins or small nucleic acids, it's usually composed of different concentrations of acrylamide and a cross-linker, producing different sizes of mesh networks of the polyacrylamide. When we are separating larger nucleic acids, the preferred matrix is the purified agarose. Now, in both the cases, the gel forms a solid yet porous material for the molecules to pass through these pores. Now, acrylamide, in contrast to polyacrylamide, is a neurotoxin and it must be handled with appropriate safety precautions to avoid any kind of poisoning. On the other hand, your agarose gel is the agarose is composed of long unbranched chains of uncharged carbohydrates without crosslinks, resulting in the gel with large pores for the separation of macromolecules and macromolecular complexes. After this we run the gel. So after the electrophoresis is complete, we need to check these gels under some kinds of stains or under some kinds of light like the UV transilluminator or so on. So the agarose gel is stained with an ethidium bromide. That can be also done when we are previously preparing the gel. For an SGS page, we go for a silver or a Comasi blue staining. Now there can be other methods as well to visualize the separation mixture. Now if we use some kind of a fluorescence molecule while making or preparing the samples that we are loading onto the gels, we can also see it under the UV light that will fluoresce, and you can take a photograph under that as well. Now, if the molecules contain some kind of radioactivity added to visibility, so an autoradiogram can also help you out, and that will help in the recording of the gel under an autoradiogram. Now, if several mixtures have initially been injected next to each other, they will run parallel in the individual lanes depending on the different molecules. Each lane shows the separation of the components from the original mixture as one or distinct bands. Now, incomplete separation can lead to overlapping bands or to indistinguishable smears representing unresolved components. Therefore, it's very important to get an idea of where our protein is, what is the size of the DNA that we are loading, because then we might need to alter the concentration of the agarose or the polyacrylamide during the preparation of the gel for the relevant or very nice resolving for better resolution of the molecules in that particular range. Now, bands in different lanes that end up in the same distance as pass through the gel with the same speed, which usually means that they are of the same size. There are molecular weight size markers. Okay, so there are molecular weight markers that are available to contain a mixture of the molecules. So the markers, the ladders all means the same. The molecular weight detectors can all mean the same. So contains an available mixture of molecules of the known sizes. If such a marker is run on one gel of the parallel to the unknown samples, it can be observed and can be compared of the unknown in order to determine their size. Now, the distance a band travels is approximately inversely proportional to the logarithm of the size of the molecule. This is very important because sometimes what happens is that, now I do not have probably a slide on this, but this line is very important. You see that you have a molecular weight marker running parallel to your protein of interest. Okay, for example, if I go over here, this is a molecular weight marker or a ladder, and this is the protein of your interest. Say it's over here. So what does it say? So the distance a band travels is inversely proportional to the logarithm of the size of the molecule. Now you know that these molecular weight markers that are running over here are of the exact size, because this is a molecular weight marker and it's of a known size. So if you take the logarithm, the inverse, the logarithm of this molecular weight, and you plot onto a gel where you have in the y-axis as logarithm of molecular weight, and in the y-axis, in the x-axis, you have the distance. So depending on the distance that it has traveled, you can actually tell whether your so the distance traveled is less over here, that means that it's of a higher molecular weight. Absolutely fine. Now as I was telling over here when I was talking, like it can be between a range of 100 to 95. We do not have to do that ideally, because we can take the distance that it has traveled. So from here we know that it has traveled of say 2 centimeter. Over here it's about 7 centimeter, but this is also about 6 centimeter, right? So when we do the logarithm, we can also tell what is the distance that it has traveled, because that's a known variable to us. What we do not know is about the size. Probably, I'm guessing we do not know the size. Then we plot the same distance onto that known reference line that we had plotted according to the molecular weight markers, and we can tell the exact weight of this particular protein. So this thing, the distance a band travels is inversely proportional to the logarithm of the size, is a very important graph which can tell you the exact size of a protein when you're running an SDS page. It's a very important question because I do not know in which fields you are or in which stages of your education you are, but in many interviews and in many exams they do ask that whether you can tell the exact molecular weight of a protein from an SDS page, and most of the people tell no. Because why is it so? Because they know that you can only tell about the approximate size by looking at the molecular weight. So 150 is there, so you know it's somewhere between 100 and 50 kilodaltons. But if you plot this particular graph where you have the logarithm of mw versus the distance, it can tell you the exact size of a protein. Or this thing is even applicable for an agarose gel electrophoresis. Okay, so this is a very important graph that you can study about. You can also do some, if you have some kind of an SDS page, or you run some kind of an SDS page in your lab, you can do it these kinds of things even in your home. Okay, so now talking about proteins, which unlike the nucleic acids have varying charges and the complex shapes, therefore they may not migrate into the polyacrylamide gel at similar rates when we place a negative to positive EMF on sample. Now proteins therefore are usually denatured in the presence of a detergent such as an SDS that coats the protein with a negative charge. Generally, the amount of SDS bound is relative to the size of the protein, that is 1.4 grams of SDS per gram of protein. And in ideally, what we see, so so if it's of a size, if a protein of size 100 amino acids is there, we require 50 molecules of SDS. So 2 amino acids can be charged by 1 molecule of SDS. We can think it in that way. Okay, now since denatured proteins act as long rods instead of a complex tertiary structure, the rate at which it moves is based on its size and not on its charge or shape. The other way, so the other kind of gel electrophoresis which you probably have heard about is the native gel electrophoresis, which is mainly used in the protein electrophoresis where proteins are not denatured. So the separation here is based on charge to mass ratio. In an SDS page, the charge is uniform. Okay, but so you are separating it based on size. But for a native gel, it's according to the charge by mass ratio, because the charge of the protein molecule is different according to the pH of the buffer that you're using, and so on and so forth. Now the other is the polyacrylamide gel electrophoresis, where you can be separating proteins ranging from 5 to 200 kilodaltons, even more than that, due to the uniform pore size provided by the polyacrylamide gel. So mostly the pore size is controlled by controlling the concentrations of the polyacrylamide and bisacrylamide used in creating a gel. Now, acrylamide, as I mentioned, is a potent neurotoxin and should be handled carefully. The other type we can use is the agarose gel, which can be used to separate native protein. So they have a uniform pore size but are optimal for electrophoresis of proteins that are larger than 200 kilodaltons. Okay. So we use a page for proteins below 200 and an agarose gel for proteins above 200 kilodaltons. Now, unlike the SGS page type of electrophoresis, the native gel electrophoresis does not use a charged denaturing agent, be it the CTAB or the or the SGS page. The molecules being separated are usually proteins, therefore the different molecular mass and an intrinsic charge and experience different electrophoretic forces dependent on the ratio of the two. Now, since the proteins remains in the native state, they may be visualized not only by a general staining but also specific enzyme linked staining, which is usually hampered when you are running a SDS page, because primarily due to two reasons. One is that the enzyme linking will not be possible in the presence of a detergent, so it hampers the affinity of these two protein-protein interaction. And secondly is that the protein is in open form, that can also lead to, so the tertiary structure might be involved, or the might be involved in the linking process, but that is usually hampered when you're opening up or denaturing an entire protein. So the SDS page is usually used in different aspects of biochemistry, genetics, molecular biology, forensics, and so on. So having the SDS page is of samples having identical charge per unit mass is due to the binding of the SDS, results in fractionation by size, and is probably the most widely used biochemical method. Okay. Now, the solution of the proteins to be analyzed is first mixed with SDS and an ionic detergent, which denatures secondary and non-disulfide linked tertiary structures and applies a negative charge to each of the protein in proportion to its mass. Without SDS, what happens? The different proteins with similar molecular weights would migrate differently due to the difference in the mass charge ratio, as each protein has an isoelectric point and the molecular weight particular to its primary structure. Now, this is known as the native page. Now, adding SDS solves this problem as it binds to and unfolds the protein, giving the near uniform negative charge along the length of the polypeptide. Now, SDS binds in a ratio of approximately 1.4 grams of SDS per 1 gram of protein, given the uniform mass by charge ratio of the proteins, so that the distance of the migration to the gel can be assumed to be directly related to only the size of the protein. So this is the general procedure which I discussed even previously. Okay. Now we should be discussing about the chemical ingredients and the roles of these different chemical ingredients that we use in our page. So for a polyacrylamide gel, it's known to be a potential embedding medium for sectioning tissues since 1954. So there were two independent groups, David and Raymond, that employed the PAG in electrophoresis in 1959. It has several electrophoretic features, making it a versatile medium and in today's generation a widely used medium for different sections of biology. Okay, so page separates protein molecules according to both size and charge. So the properties of the SDS page gel is that it's synthetic gel, it's thermostable, it's transparent, strong, and it's also chemically inert, and can be prepared with a wide range of average pore sizes. By how do you change the pore sizes? So the pore size is determined by two factors. One is the amount of acrylamide present and the amount of crosslinker. So the total amount of acrylamide present, that is percent T, is the total acrylamide bisacrylamide monomer concentration, and the amount of crosslinker or percentage C is the crosslinker concentration. Now the pore size decreases with increasing percentage of T with cross-linking. Now 5% C gives the smallest pore size. Any increase or decrease in percent C increases the pore size, as the pore size with respect to percent C is a parabolic function with vertex as 5% C. Now this appears to be because of the non-homogeneous bundling of the strands in the gel, right? Now this type of gel material can withstand high voltage gradient. So it's made in such a manner that it can withstand high voltage gradient feasible for various staining purposes and destaining procedures, and can be digested to extract separated fragments and dried for autoradiography, permanent recording. Sometimes people want to do mass spectrometry by destaining that portion of the gels. You can do all of that. And these particular types of gel, the SDS page has two layers of gel. One is the stacking or the special gel for the stacking of the protein fragments, and the other portion is the resolving or the separating gel for the separation of the molecules. Now the different chemical ingredients present over here is tris, that it is being used as a buffer because of its innocuous substance to the proteins. It has a pK of 8.3 and it makes it a very fantastically workable buffer in the pH range of 7 to 9. The other component is glycine, which is being used as a source of a trailing ion or a slow ion because of its pK in the range of 9.7, and its mobility of the glycinate is such that the effective mobility can be set to a value of the slowest known protein of net negative charge in a pH range. Now, not only this is the role of glycine, rather glycine has a very different role when it is in a stacking gel and when it is in a resolving gel. Okay, so in a resolving gel, we use a tris that is of a pH of 6.8, and in a resolving gel we use a pH of 8.8. So here the glycine would have two different charges, right? So in stacking gel, what happens is that you have, so there are the role of 3 ions present over there, and in the resolving gel also there are 3 ions present over there. So in the stacking gel, what happens, you have the glycine that is in a neutral charge because it's of a pH 6.8. So in that particular pH, glycine would be in a zwitterionic form. The other ion that plays a role over there is the chloride ion. That's the smallest ion with the highest amount of charge, so its mobility will be very fast. So chloride moves the fastest. In between this chloride and the negatively charged protein, okay, the even slowest mobility is shown by the glycine that is of a neutral charge. So the protein is packed between the fast moving chloride ion and the slowest moving glycine. Okay, so that is why in between the two ions is stacked, the protein gets stacked, and that is important. That is the role of the stacking gel over there, for which a trace of pH 6.8 is used. Now when you move to the resolving gel, the pH changes to 8.8. At that particular pH, glycine is negatively charged and it becomes glycinate. Okay, so glycine is negatively charged, but it does not move as fast as your chloride. So chloride, there also it moves faster, but the places are changed between the protein, the negatively charged protein, and the glycinate. So after chloride moves your glycinate and then your proteins. That allows for the resolving of the proteins according to their mass. Okay, so that is why glycine is a very important ion in SDS page. The other factor is your acrylamide. So it's a white crystalline powder. Now when you dissolve it in water, it autopolymerizes. Now it's a very slow spontaneous process by which acrylamide molecules joins together via the head to tail fusion. But in the presence of free radicals, the process becomes faster, and this forms long chains of polymers that forms the gel matrix. Now this is known as vinyl addition polymerization, the name of the reaction. Now a suitable solution of this polymer is also available that becomes viscous but does not form a gel, but the chain simply slides over one another. Now when you use cross linker or something, the gel formation actually occurs. Acrylamide is a neurotoxin, so it's essential for the acrylamide to be stored in a cool, dark and dry place to reduce any sort of autopolymerization and hydrolysis. Now we have the bisacrylamide, that is also a type of an acrylamide. Okay, so it's used as a cross-linking agent for the polyacrylamide gels. So the two acrylamide molecules are coupled head to head at the non-reactive ends, as it acts as a cross-linking agent for the proper pore formation over there. The other important factor is your SDS, or the sodium dodecyl sulfate. So it's the most common dissociating agents used to denature native proteins to individual polypeptides. Now when a protein mixture is heated to 100 degrees in the presence of SDS, the detergent wraps around the polypeptide backbone, the protein opens up, and the SDS molecules can go and sit over there, giving it a constant, giving it a uniform negative charge to the protein. That's majorly contributed by this SDS. Now this polypeptides after treatment becomes rod-like structures possessing uniform charge density with the same negative charge per unit length. But who initiates this gel formation? For there, we have the ammonium persulfate, or the APS. That's an initiator for the gel formation. What does it initiate? It initiates, like I was telling over here, if we have the, where was it? In the presence of the free radicals, the polymerization process just speeds up. So the ammonium persulfate, or the APS, initiates the sulfate or the sulfide free radicals, and these free radicals that are generated are important in the fast formation of the gels, or the speeded up polymerization process of this particular matrix formation, right? Okay, so the other one is timid. So we have the initiator, that's APS. We have the TIMET. So that's used. You can see. Okay. So TIMET. So the chemical polymerization of the acrylamide gel is used for SDS. It can be initiated by ammonium persulfate or the quaternary amine. TIMET. So the rate of the polymerization and the properties of the resulting gel depends on the concentration of APS and TIMET, lowest catalytic concentrations that allows polymerization in the optimal period. So your APS is the initiator and TIMET is the stabilizer. So what does it stabilize? Now after the formation, so we know that free radicals have the highest entropy. So the joining and the breaking of the bonds formed between the free radicals are very spontaneous. Okay, and they are entropically, these bonds are very entropically high. Okay, so it stabilizes. So once a bond is formed, TMED is involved in stabilizing this bond such that it does not again go back to its, it does not again break up. Okay, so that is where the role of TMED comes. It stabilizes the formation of the bonds, thereby speeding up the polymerization process. Otherwise, it will take a longer time for the gel to ultimately form. So that is why APS and TMED is very, very important, and that also in a certain concentration. You cannot use large amounts of TMED or APS, because then before you start loading the gel for its preparation, it ultimately forms a gel-like material even in the falcon in which you are preparing the gel. Okay, so you need an optimum amount of time, the optimum amount of concentration of all these chemical ingredients in the gel formation. Okay, now Now after this you need to run a gel, you need to stain the samples, and you need to stain the gel for observing the proteins present over in the molecules in the gel after you've run it. So for that you have two stains over here, the bromophenol blue. So it's used as a universal marker dye for staining your samples. So you need to stain the samples because you cannot load the um, transparent proteins onto your gel. You need to stain the samples with something such that you can understand till how much has your gel run, or your proteins, how well you want to dissolve your protein, how long you want to run your gel. So proteins and nucleic acids are mostly colorless when they are separated to electrophoresis. It's important to stop the run before the gel, before the run of the gel. So it's important because to stain it using a tracking dye. So bromophenol blue is our tracking dye because it's viable in alkali and neutral pH, and it's a small molecule, so it runs the fastest ahead of all the protein molecules that are usually of higher sizes. Now, being a small molecule, it moves ahead of most the proteins and nucleic acids. As it reaches the ionic end of the electrophoresis medium, is stopped, and it can bind with proteins weakly to give it a blue color. So the color is attained, but it does not react with the protein. Okay. The other dye that we use is the Comasi Brilliant Blue. So it's a popular protein stain that is after the gel is run. We stain it using the Comasi Blue's Brilliant Blue. It's an anionic dye that binds with proteins non-specifically, and the structure of CBB is non-polar. So we use in the presence of a methanolic and in the presence of methanol and acetic acid. So the proteins are fixed by acetic acid and are simultaneously stained. Now we can remove the excess of the dye. We wash it with water and then we add a destaining solution. So the difference between a staining and the destaining solution is that you have methanol, acetic acid, water. In the staining solution you add this some amount of Comasi Brilliant Blue powder, but in the destaining solution you just do not add the powder. Otherwise all the ingredients are same. So the proteins are detected as blue bands on a clear background after the destaining process, and as SDS is anionic, it can interfere with the staining process. Therefore, a large volume of staining solution is used. But we are using a methanol solution. So methanol is basically used to stop the activity of the SDS from any kinds of reaction or such. But in any ways, we use large amounts of staining solution um, for a better staining process. Okay. So that's all about the SDS page or a gel electrophoresis and an agarose gel electrophoresis. I guess will be covering most of the questions that might be asked in the different exams and all. The later portions are very, very important, where the roles of the chemical ingredients are there, the basic principles before that's we have for a gel electrophoresis and all, and maybe a few here and there questions can be there depending upon the size, the molecular weights and all. Okay, so maybe 2 to 3 minutes of a break, after which we can discuss the practice problems. Okay, in the meantime, if you have any questions, you can ask me in the chat box. I will be happy to answer them. Okay, we will be discussing the problems in a few minutes. Right. Okay, so let us start with the practice problems. You people can answer me. Um, you can start with the answering, and then probably I will be telling you. These are you can expect pretty easy questions from here on. Okay, so right, in gel electrophoresis, the gel acts as molecular glue, molecular copy machine, molecular sieve, molecular scissor. Using molecular glue. No, does it actually act as a molecular glue? Yeah, right. So it's a molecular sieve. Okay, so it acts as a molecular sieve over here. Okay, right. So which of the suitable techniques for separating large DNA fragments? So what do you use for separating large DNA fragments? A. Yeah, absolutely right. So ASDS polyacrylamide gel is used for a protein. Native polyacrylamide gel is also not used. That's also used for a protein. So agarose gel electrophoresis is definitely correct over here. So which of the following can be determined using SDS page? Molecular weight of a protein, PI of a protein, presence of disulfide bonds in a protein, dissociation constant of protein ligand complex. B. Okay. So the SDS page, so that means the protein is denatured. You are separating it based on the molecular weight. So the first one is definitely correct, because the molecular weight of a protein is what you exactly see in a SDS page. Now the PI of the protein cannot be determined because you are not running a native page where you are separating according to the charge to mass, right? Now, when you're running a native page, you can separate it according to its PI, because if you're using a positively charged buffer, a buffer of higher pH, okay, then your protein would be negatively charged. But if it's lower than your PI, it will be positively charged. So on the basis of that, you can do it, but that's for a native page. The last one, dissociation constant, cannot be set from an SDS page, because in any ways you are say opening up the proteins, you're denaturing the proteins, so the kd value cannot be found out. But the presence of disulfide bonds in a protein can be definitely be found out. How is it so? Because if your sample does not have BME, so what is BME? It's a reducing agent. So if there is no reducing agent present, then the disulfide bonds would be intact, and you would be observing a dimer or a trimer or maybe a tetramer present over there if it's an inter or an intramolecular disulfide bond. But if it's not, when does it happen? If you are using a reducing agent, then the disulfide bonds won't will break up, because you are using a reducing agent, and you will be observing only one band. So in the presence and absence of a reducing agent, there is a difference in the bands. So because of that, a molecular weight of the protein as well as the disulfide bonds in a protein can both be determined. Correct? Okay. Now what is the role of SDS in SDS page? Absolutely correct. It imparts uniform negative charge to the proteins. Migration of a protein in SDS page is determined by charge of the proteins, size of the protein, shape of the protein, or PI of the protein. I just said it. SDS page, or you just answered it. SDS page, in an SDS page, you have a uniform negative charge. So definitely you're not going to separate it according to this charge. If you would have done that, all the proteins would be in the same row or in the same place. So you are separating it based on the molecular weight. So that's size. Okay, so you're separating it based on the size of the protein. So B is the correct option over here, not even shape or PI, right? The next one. So B is the correct option, not A. The main advantage of using separate gels for stacking and resolving in SDS page is to increase the resolution of separation, increase the migration rate, maintain the charge of the proteins, retain the shape of the protein. I just explained about stacking and the resolving gel. So why is it, what's the main advantage behind it? No? No answer? Okay, so let's see from the last. To retain the shape of the protein, that's definitely not true because you're running an SDS page. To maintain the charge of the protein, that's also not true because you're running it in an SDS page. So you cannot retain the shape. You cannot even maintain the charge because it's already maintained. You're having a uniform negative charge over here. To increase the migration rate is increased whether the size is small or the size is low, or if you are changing or manipulating, modulating the power supply. That's also not true. But to increase the resolution of the separation, because in the stacking you stack the proteins between tris and between chloride and glycine, but in resolving you have chloride running fastest, then you have the glycinate which is running the second fastest, and then your proteins that start resolving according to the size or the molecular weight. Okay, so option A is correct. Next one. The role of APS in SDS page or native page is, so you have to tell about the role of ammonium persulfate. Catalyze polymerization of acrylamide, cross-link acrylamide and bisacrylamide, provide free radicals for cross-linking reactions, and to maintain the pH of the gel. Yeah, you're partly correct over here to cross-link acrylamide and bisacrylamide, but there is something else that has a more so there is a major role of APS more than the cross-linking. So it provides the free radicals for the cross-linking of this particular reaction. So firstly, it has to provide the free radicals. That is what it does. The cross-linking process is generated through the free radicals, but APS does not do the cross-linking. The free radicals does it. So APS generates the free radicals. The free radicals does the cross-linking. Okay. So the major role of APS is to generate the free radicals. Cross-linking happens through itself by the free radicals. Okay. So option C is a more perfect of an answer. Next one. In SDS page, the major ions present in the buffer are tris, glycine, chloride, all of the above. I was just speaking about the role of the ions in an in the stacking and the resolving gel. So which of them are there? Tris, glycine, chloride, or all of the three? Yeah, absolutely. So you have tris in the buffer in the running buffer over there, as well as in the stacking and the resolving gel, you have the glycine and the chloride, the role of which I just mentioned. So all of these ions are present over there. The next one, BME or the beta mercaptoethanol is used in SDS page to break hydrogen bonds and unfold a protein, break disulfide bonds, break the peptide bonds, break multimeric proteins into monomers. So what is the role of BME? Break the disulfide bonds. Absolutely correct. Because it's a reducing agent, it reduces the disulfide bonds between the cysteines, right? Okay. Now agarose is, what is an agarose? Protein, polysaccharide, DNA, or a lipid? Yeah, it's a polysaccharide. That's absolutely correct. Okay, so these are easy questions. Next. Okay, the non-essential component of SDS page. So one of the component is not essential. Essential does not mean it's not required, but sometimes you can skip it. It means that. So non-essential component in SDS page is acrylamide, tetramethyl ethylene diamine, that is the TMED, ammonium persulfate APS, and the beta mercaptoethanol BME. So which among the four is a non-essential component? Okay, so polyacrylamide is not important over here. No, that's not correct. You require a polyacrylamide or a bisacrylamide for the formation of the mesh-like structure or the gel-like formation. How can you skip an acrylamide? Though acrylamide and bisacrylamide are in a specific ratio, but acrylamide is an essential component. Even you require a TMAD and APS. What you don't require always is the beta mercaptoethanol. Why is it so? Because if you want to check whether your proteins have a disulfide linkage or not, you cannot add a reducing agent over there, or you cannot add BME over there. So it's not essential, but most of us use it because we want to see the protein in its monomeric form and like that. So the reducing agents are not essential or important in the gel preparation, but it's recommended to provide it in the samples to break the disulfide bonds. Correct? So beta mercaptoethanol is not an essential candidate. The next one, to visualize proteins in gel. So we want to visualize proteins in gel. Which of the following staining method is used? Both A and D. Okay. Yeah, that's absolutely correct. So the silver staining and Comasi blue staining. Correct. In native page, so now we are coming to native page. In native page, the proteins separate depending on their size, charge, pi, all of the above. Absolutely correct. Right. So in native page, you do not have an SDS present over there or a denaturing agent. For that, you can separate it according to its charge. The size always remains over there in electrophoresis, and even the PI, because the buffer, the pH of the buffer will determine whether your protein will be positive or negatively charged. And depending on that, you have to interchange the plug points sometimes for running the gel in an opposite direction. Okay, the pore size of a polyacrylamide gel. So how can you change the pore size of an polyacrylamide gel? Changing the acrylamide concentration, changing the bisacrylamide concentration, changing the ratio of acrylamide and bisacrylamide, all of the above. Okay, so D, yes. So you can change all of the above in different ways to change the pore size of an acrylamide gel. That is correct. In electrophoresis, DNA will migrate towards anode, cathode, depends on DNA sequence, depends on DNA length. Very easy. What happens in electrophoresis? DNA will migrate towards. Yeah, DNA is negatively charged. It moves towards the anode. Absolutely. Okay. Now, to reduce the pore size of agarose gel, what do you need to do? Increase agarose percentage, decrease agarose percentage, increase salt content, change the pH of buffer. Exactly. To reduce the pore size, you need to increase the agarose percentage. Very good. So A is correct. Okay. Now the next one. Which of the following statements is true? In SDS page, all proteins are negatively charged. In native page, proteins can be negatively charged, positively charged, or neutral. In agarose gel, nucleic acids are positively charged. No denaturing agent is added in agarose gel electrophoresis. A. Okay. In SDS page, all proteins are negatively charged. Yes, that is right. Anything else? Yes, other things are also right. In native page, all proteins can be negatively charged, positively charged, or neutral depending on the pH of the buffer as well as the pi of the proteins. So A and B is correct. In agarose gel, nucleic acids are positively charged. That is incorrect. And also, we do not use any denaturing agent in an agarose gel electrophoresis. So A, B, D, all three are correct. Anyways, okay. Which of the following is true? A polyacrylamide gel can be depolymerized by heating. An agarose gel can be depolymerized by heating. In the absence of bisacrylamide, acrylamide forms a linear polymer. It is easier to control pore size in polyacrylamide gel than agarose gels. D. Okay. Okay. Anything else? Yeah, this is absolutely correct over here. Anything else? An agarose gel can be depolymerized by heating. That is not right. An agarose gel can be depolymerized by heating. That is correct. In the absence of bisacrylamide, acrylamide forms a linear polymer. That is also correct. So here B, C and D, all three are correct. Okay, so the last set. To visualize DNA in a gel, the common dye used is Pomasi blue, ethidium bromide, bromophenol blue, all of the above. Very easy. Yeah, absolutely. We use ETBR for staining, for visualizing of DNA. And the last question, in agarose gel, the migration of DNA depends on. shape, size, net charge, agarose concentration in the gel. Size, that is correct, but also shape, it matters. Why? Because I was telling what happens in the case of a plasmid. What is a plasmid? It's a supercoiled DNA. Okay, so when you are loading, when if you ever load a plasmid onto a DNA gel, a freshly isolated plasmid, you also see three bands over there. Why? Because the plasmid is in linear form, circular form, and a supercoiled form, and all of the three runs in three different directions, although you have only one plasmid over there. It's the different orientation, the different shape that matters. Okay, so shape and size both are correct over here. Okay, so that's all about the question and answers. Okay, if you have any question, you can ask me. If not, we can end the class for today. So I was busy in something on Friday, for which I could not take the class on Friday, but usually I would be taking classes on Friday only, so I won't bother you on Monday. uh, so it's just because I was busy into something. Okay, so the next class will happen on this Friday this week. Okay, on the week 7th thing. Any questions? No. Okay, then thank you for joining the class. We will again meet in the next week with a different set of lectures and question and answers. Okay, then. So thank you and bye.